Hello, my name is Gabby, and my group, which includes Katie and Como, have done our presentation on the modulation of movement by the basal ganglia. Here we will dive into the function, purpose, anatomical structures, pathways, and disorders that affect the basal ganglia.
The basal ganglia is located deep inside the brain in the cerebral hemisphere, close to the base. The main purpose of the basal ganglia is motor control. It is important to note that although the basal ganglia regulates upper motor neurons, it does not directly influence lower motor neurons. To help achieve its purpose, to put it simply, the basal ganglia has information from the cortex inputted, then outputs the information to the motor centers. Lastly, it then returns the information back to the cerebral cortex. This is just a short overview of what the basal ganglia does. We will get more into the details during our presentation.
There are a few anatomical components of the basal ganglia. The dorsal striatum, or simply the striatum, consists of two parts, the caudate and putamen. The parts of striatum are separated by the internal capsule, whose myelinated fibers radiate through striatum, giving it a characteristic striped appearance. The putamen is a round structure situated at the base of the forebrain. It is most lateral of the basal ganglia on the axial section of the brain. The main function of the putamen is to regulate motor functions and influence various types of learning, and it employs dopamine to perform its functions. The caudate lies anterior to the thalamus. The caudate integrates sensory information about the spatial position of the body, and according to that, it sends the information about the necessary fine-tunes of the motor response to that stimuli to the thalamus. Additionally, it contributes to body and limb posture and the speed and accuracy of directed movements. Besides motor control, the caudate is involved in many other tasks, such as memory, goal pursuit, learning, language, processing, and emotions. Subthalamic nuclei lies in the ventral thalamus. Substantia nigra is in the base of the midbrain. The substantia nigra and subthalamic nuclei are closely associated with the motor functions of the basal ganglia. These two comprise a subcortical loop that links most areas of the cerebral cortex with upper motor neurons in the primary motor and premotor corticals in the brainstem. Their influences on upper motor neurons are required for functional regulation of voluntary movements. The globus pallidus and the substantia nigra are the main sources of output from the basal ganglia complex to other parts of the brain. The main output is inhibitory. The substantia nigra contains important dopaminergic cells. The major input to the basal ganglia is to the striatum. Inputs come from the cortex and the brainstem. The substantia nigra is made up of two distinct regions, the substantia nigra pars compacta and the substantia nigra pars reticulata. The substantia nigra appears in the brainstem that looks like a dark streak in unstained brain tissue. Its appearance is where it gets its name, as substantia nigra is Latin for black substance. Substantia nigra is shown in the picture as the green-colored portion at the bottom. It contains a large number of dopamine neurons and high levels of a dark pigment called neuromelanin. Most of the dopamine-producing neurons of the brain are found in the substantia nigra. The destinations of incoming axons from the cerebral cortex are the dendrites of a class of cells in the striatum, called medium spiny neurons. The large dendritic trees of these neurons allow them to collect and integrate input from a variety of cortical, thalamic, and brainstem structures. The axon arising from the medium spiny neurons converge on neurons in the pallidum, which includes the globus pallidus and the substantia nigra pars reticulata. Medium spiny neurons in the striatum discharge prior to movement initiation and at termination. Thus, the basal ganglia is implicated in the selection of action plans and execution of habitual movements. Output mainly comes from the globus pallidus and goes to the cortex by the thalamus. The main output response is inhibitory.
The role of the basal ganglia in action selection is complicated due to its involvement in disinhibition. The direct pathways initiate movement, while indirect pathways inhibit or terminate movement. The direct pathway includes excitatory signals such as glutamate that are sent to the striatum from the motor cortex. The striatum then sends inhibitory neurons such as GABA to the globus pallidus internal segment and the substantia nigra. Then the globus pallidus and substantia nigra send inhibitory neurons to the thalamus, which then sends excitatory neurons back to the motor cortex. Thus, the direct pathway allows for disinhibition of thalamic control and motor planning. For the indirect pathway, it starts the same with excitatory signals sent to the striatum from the motor cortex, which then sends inhibitory signals to the globus pallidus external segment. The external segment of the globus pallidus will send inhibitory signals to the subthalamic nucleus, which will then send excitatory signals back to the globus pallidus internal segment and the substantia nigra. The same ending of the pathway occurs, which includes the globus pallidus and the substantia nigra sending inhibitory neurons to the thalamus, which then sends excitatory neurons back to the motor cortex. The indirect pathway allows the activation of inhibition, which turns off the motor cortex. In this figure, we can see the pathways of neurons and interneurons through the specific structures that were described in the previous slide when examining both direct and indirect pathways.
Dopaminergic neurons have a big influence on the cortical input. Direct pathways have a D1 receptors. These receptors activate and excite when they receive dopamine from the medium spiny neurons. Indirect pathways have a D2 receptors. These D2 receptors inhibit and suppress when they receive dopamine from the medium spiny neurons. Dopamine is important for motor control, as well as it helps us have a reward response, which is great for creating habits. Parkinson's disease is a hypokinetic movement disorder. It is a degenerative disease of the nervous system. It is mainly caused by the loss of nigrostriatal dopaminergic neurons due to a large restriction of substantia nigra pars compacta. This restriction decreases dopamine, which is important for us because it creates reward, wakefulness, transmission of messages, and most important, control of body movements. The lack of dopaminergic neurons creates a decline in motor control, which can progressively get worse. This is an image of the input and output of the basal ganglia of someone with a hypokinetic movement disorder. It is important to look at the substantia nigra pars compacta. You can see the arrows are fading to show the decrease in dopaminergic input. This increases inhibition from the pallidus to the thalamus, which makes it less likely for the thalamus to excite the motor cortex. Huntington's disease is a hyperkinetic movement disorder. It shows up most commonly as a mood alteration, but it is most known for involuntary movements. but there is no weakness or deficient of sensory function. Involuntary movements from this disease is caused by an insignificant tonic output from the pallidus. Insignificant output is caused by degeneration of medium spiny neurons. The medium spiny neurons have an inhibitory input that is not lessened, which creates the globus pallidus to be very active, which reduces the inhibitory outflow from the basal ganglia. This image is showing the reduction in the Huntington disease causes to the putamen and the caudate. You can specifically see this reduction by looking at the red arrows. This image is showing the input and output of the basal ganglia. If you take a look at the arrow from the caudate and the putamen to the pallidus external, it is labeled as degenerative, showing the effects of hyperkinetic disorders.
This figure shows the differences among the motor and non-motor loops that affect the basal ganglia. The disruption of these loops that cause damage are likely to develop cognitive disorders in addition to Huntington's disease and Parkinson's disease, such as Tourette syndrome, schizophrenia, obsessive-compulsive disorder, depression, and chronic anxiety, which result in the disruption of these specific loops. Here we have our 5 multiple-choice questions. First question is, which part of the basal ganglia contains dopaminergic cells? Is it A, globus pallidus, B, substantia nigra, C, caudate, or D, subthalamic nucleus? Our second question is, what are the 2 components of the striatum? We have A, the caudate and lunate, B, the pallidum and caudate, C, the globus pallidus and putamen, or D, the putamen and caudate. Our third question is, what is the predominant excitatory neurotransmitter of the basal ganglia? Our options are A, dopamine, B, GABA, C, glutamate, or D, acetylcholine. For the fourth question, we ask, what is the predominant inhibitory neurotransmitter of the basal ganglia? Is it A, dopamine, B, GABA, C, glutamate, or D, acetylcholine? And for our last question, we ask, which of the following are considered hyperkinetic disorders? Is it A, Huntington's disease, B, chronic anxiety, C, depression, or D, Parkinson's disease?